I want you to picture something for me. And you can close your eyes if you're not, driving or operating heavy machinery.
Hey, good disclaimer.
Imagine a factory, but not just any factory. This is the most complex, the most high-speed manufacturing plant in the entire universe. It is running 24/7.
No holidays.
No holidays, no coffee breaks. It's just constantly churning out proteins, burning fuel, repairing its own heavy machinery. I mean, it is loud, it's hot, it is industrial.
And it is messy work.
Yeah.
That's the part that I think people forget. Manufacturing always creates waste.
Incredibly messy. You've got smoke, you have scrap metal piling up in all the corners, you've got these toxic byproducts just leaking onto the concrete floor. Now, in a normal, well-run factory, the night crew comes in.
Right.
The janitors, the maintenance team. They sweep the floors, they fix the broken gears, they haul all the trash to the incinerator.
Maintenance is everything. I mean, without it, production is completely irrelevant.
Exactly. But now imagine for a second that the night crew just never shows up.
Oh, that is a disaster just waiting to happen.
The trash just piles and piles up. The gears start to grind because there's grit in the oil now. The aisles get blocked with all this debris so the workers can't even get to their stations.
And eventually the whole operation it stops. It just collapses under the weight of its own waste. It becomes toxic.
That scenario, that isn't just a metaphor for a bad business model. That right there is basically the fundamental definition of aging. That is what happens to our bodies when a specific process stops working.
We're talking, of course, about autophagy.
Autophagy. It sounds like a death metal band or maybe a sci-fi villain. I am Autophagy, destroyer of worlds.
It does have an intense ring to it, doesn't it?
Okay.
But the etymology is actually, well, it's kind of poetic in a biological way. It comes from the Greek. You have auto, which means self.
And phagy, which means eating, self-eating. Which, honestly, when I first read that in the research packet for this, I recoiled a little bit. It sounds destructive. It sounds like something you actively want to avoid. Like, why would I want my cells to eat themselves?
It's so intuitive to think that, right? We're biologically programmed to think eating yourself is a bad thing.
Yeah.
But in biology, destruction is the absolute prerequisite for renewal. You cannot build the new without first dismantling the old.
You can't renovate a house without tearing down the rotting drywall first.
You can't. And that is really the mission for this deep dive. Because autophagy has become this massive, massive buzzword. I see it on Instagram. I see it on supplement bottles. People say things like, oh, I'm skipping breakfast today to activate my autophagy, as if it's just some kind of simple turbo button you press to burn fat.
Yeah, like a cheat code in a video game.
Exactly like a cheat code.
Exactly.
Yeah.
But the reality is so much more complex and, frankly, so much cooler than the sales pitch. We aren't just going to tell you that fasting is good. We are going to open up the hood of this cellular recycling plant. We need to understand the gears, the levers, and this is the most important part, why this whole system starts to fail as we get older.
And we have so much to cover.
Yeah.
I mean, this isn't just about dieting. This is an evolutionarily conserved process.
What does that mean, conserved?
It means it's ancient. We see it in yeast, we see it in worms and fruit flies, and obviously in you and me. We share this fundamental mechanism with almost every eukaryotic organism on Earth.
So the blueprint hasn't really changed much in millions and millions of years?
Remarkably little. We've identified over 32 specific genes, they're called the ATJ genes for autophagy, that orchestrate this whole dance. So whether you are a microscopic yeast cell starving in a petri dish or a human being running a marathon, the machinery that kicks in to recycle your cellular parts is largely the same.
Wow.
It's the primary way life balances its energy budget during times of stress.
So, our goal today is to move beyond the buzzwords. We are going to break down how this machinery actually works. We're going to look at the double-edged sword risks because, spoiler alert, autophagy is not always the good guy, especially when we start talking about cancer.
That is such a crucial nuance. And we'll also dig into the Pearl trial. This is a major new study on longevity in a drug called rapamycin. And there were some truly shocking findings in there about bioavailability that really anyone buying supplements needs to hear.
And then we're going to talk about the future, how we can actually measure this stuff in your own blood. Because right now, let's be honest, most of us are just guessing.
We're moving from, I think I'm fasting enough to, I know what my cellular recycling rate is.
Yeah.
And that is the holy grail.
Okay, let's unpack this. We've defined it as self-eating, but the source material you sent over highlights that it's not just one single process. It's actually more like a suite of different recycling programs. It's not just one type of garbage truck.
That's a great way to put it. We tend to lump it all together in casual conversation, but biologically, there are three distinct engines or modalities of autophagy. They all have the same final destination, a lysosome, but they take very, very different routes to get there.
And the lysosome, that's the incinerator, the cellular furnace.
Think of the lysosome as the recycling center and incinerator combined into one. It's this membrane-bound sac, and it's full of acid and these incredibly powerful enzymes called cathepsins. It's designed to shred everything down to its basic reusable building blocks: amino acids, fatty acids, sugars.
Okay, so we have the incinerator. What are the three different ways we get the trash into it?
Okay, so the first one, and the one we usually mean when we're talking about longevity or fasting, is macrotophagy. This is the heavy lifter.
The dump truck.
The dump truck, exactly. In macrotophagy, the cell builds a brand new double membrane structure called an autophagosome from scratch. Just imagine a flat sheet of membrane that starts to curl up and physically engulfs a whole chunk of the cytoplasm.
So it's not selective, it just grabs whatever's there.
Pretty much. It'll grab old, worn-out mitochondria, clumps of misfolded protein, bits of ER, whatever's lying around. It's like taking a giant trash bag and just scooping up everything on the floor.
Okay.
It seals all of that inside this vesicle, that's the bag, and that bag then travels along these little tracks inside the cell, the cytoskeleton, to the lysosome, and it fuses with it. The bottom drops out, and all the trash just falls into the acid to be broken down.
Okay, so that's the main one. That's the big one. What's number two?
Number two is microautophagy. This is the direct approach. There's no middleman, there's no trash bag.
Yeah.
The lysosome itself just folds its own membrane inward. It's a process called invagination and just grabs a little bite of the cytoplasm directly.
So it just slurps it up.
Essentially, yes. It's really useful for non-selective, smaller cleanup jobs. It's kind of like just wiping a crumb off the table directly into the bin. Much less involved.
And the third one, this one sounded the most exclusive, the chaperone-mediated one.
Right. Chaperone-mediated autophagy, or CMA for short. And I love your analogy for this. I call it the VIP entrance to the nightclub.
Okay.
It is highly, highly selective. Microautophagy, like we said, grabs big chunks of whatever. CMA doesn't do that. It looks for very specific proteins that have a particular ID card.
An ID card, you mean like a barcode on a protein?
Exactly, a molecular barcode. It's a specific sequence of amino acids known as the KFEQ-like motif. If a protein has that specific sequence exposed, it basically means it's damaged or unfolded and needs to be destroyed.
So if you don't have that ID, you're not getting in.
You are not getting in. A chaperone protein called HSC70 acts like the bouncer. It recognizes that motif, grabs the flagged protein, and walks it right up to the lysosome. But instead of being engulfed, it's fed through a specific dedicated gator receptor called AMPK2A.
So it threads it through.
It's threaded through the membrane like a string through a needle. It's incredible precision. It's not just burn everything. It's, we need to get rid of this specific protein right now.
That's amazing. And that precision must be vital for certain functions.
It's absolutely vital. But for today, for our purposes, we're mostly going to focus on that first one, macroautophagy, because that's the one that is most responsive to our diet, our lifestyle, and the longevity drugs we're going to discuss later. It's really the one that handles the bulk spring cleaning.
Okay, so let's get into the mechanics of macrotophagy then. If this is a factory, who is the foreman? I mean, who decides when to clean and when to work? Because presumably we don't want to be eating ourselves constantly or we just disappear.
Right. It's all about balance. And the cell manages this with this constant high-stakes tug-of-war between two master regulators.
I love a good rivalry. Who are the players?
Okay, so on one side, wearing the growth jersey, you have mTORC1. That stands for the mechanistic target of rapamycin complex 1. Just think of mTOR as a party animal.
A party animal.
An absolute hedonist. When nutrients are abundant, so when you've just eaten a steak or had a sugary drink, mTOR is active. It senses those amino acids. It senses the insulin, and it screams, "We are rich. Build new cells. Divide. Grow. Ignore that mess in the corner. Just keep building."
So mTOR is the anti-recycler. It's the signal for boom times.
Exactly. When mTOR is active, it physically locks the recycling crew out of the building. It does this by chemically modifying the autophagy machinery, specifically a group of proteins called the ULK complex, to keep them inactive.
Okay, so it puts the brakes on them?
It puts the brakes on, and it does something else. It also locks the master planner, a transcription factor called TFB, out of the nucleus.
That's the architect.
TFE is the guy who holds the blueprints for building new lysosomes and other recycling components. mTOR keeps him stuck out in the cytoplasm so he can't get into the nucleus to turn on the genes that build more recycling centers. mTOR basically says, we don't need trash cans, we need party streamers.
Which makes perfect sense from an evolutionary perspective, right? If you
Right.
just killed a mammoth, you don't want to be eating your own muscle tissue. You want to use those resources to grow.
Precisely. But here is the problem. We, in the modern world, live in a world where the mammoth hunt never, ever ends. We are constantly feeding mTOR with snacks, with lattes, with 3, 4, 5 meals a day.
So who is pulling on the other side of that rope? Who is the one that tells mTOR to shut up?
That would be AMPK, the energy sensor. If mTOR is the party animal, AMPK is the stern, responsible accountant,
The buzzkill.
the necessary buzzkill,
Yeah.
the responsible one. AMPK's job is to monitor the ratio of ATP, which is cellular energy, to AMP, which is depleted energy, inside the cell. When that fuel tank gets low, so you have high AMP, low ATP, AMPK wakes up, it sounds the alarm, it signals an energy crisis.
And what does the accountant do when there's a crisis?
It pulls the fire alarm. It does two critical things at the same time. First, it slams the brakes on MTR. It says, stop growing, we cannot afford it, party's over.
Right.
Second, it directly activates that ULK complex we mentioned, the one mTOR was suppressing. It flips the on switch for autophagy.
So this is why fasting is the most famous trigger for autophagy. You stop the incoming food, the energy levels drop, AMPK wakes up, it suppresses mTOR, and the factory finally starts recycling its old parts to make fuel.
That is the fundamental logic right there. And once that ULK complex is turned on, it moves to a very specific spot on another organelle, the endoplasmic reticulum, to a place called the omegasome.
The omegasome, that sounds like the end of the world, or a really intense techno club.
It does, but it's actually the beginning of renewal. This is the spot where the assembly line starts. The cell starts to recruit lipids and proteins to build that double membrane I mentioned. This is the nucleation phase. It's driven by another complex that involves a really important protein called Beclin-1 and another called VPS34. They create a landing pad using a special lipid called PI3P.
So they lay the foundation, and then it has to grow, right, to wrap around the trash.
Correct. That's the expansion or elongation phase. And this is where we see one of the most famous markers in this entire field, a protein called LC3. The cell uses two ubiquitin-like systems to stitch this membrane together and make it bigger. One of them takes a protein called LC3I and attaches a little lipid tail to it, turning it into LC32.
LC32, okay, I see that mentioned in every single paper in the notes. Why is it so important?
Because that lipid tail causes it to get stuck in the membrane of the autophagosome. It becomes part of the structure, and it stays there until the very end of the process. So when scientists want to know if autophagy is happening, they measure how much LC32 is present in the cell. It's the standard tag.
So more LC32 means more trash bags being made.
In theory, yes.
Okay.
So we'll come back to a big caveat on that later, but for now, yes. So the bag is formed, the trash is inside. What happens next?
It's got to get to the incinerator.
Exactly. Fusion and degradation. The finished vesicle, the autophagosome, is loaded onto those microtubule tracks we mentioned, and little motor proteins literally drag it to the lysosome. They fuse together, the acid and enzymes pour in, and everything inside is broken down.
And those raw materials, the amino acids and so on, are then released back into the cell to build new proteins or just to be burned for energy. It really is the ultimate circular economy.
It absolutely is. And when that system works, you have a healthy, resilient cell. But when it fails, that's when we start seeing the major diseases of aging. And nowhere is this more critical and, frankly, more terrifying than inside the brain.
Which brings us perfectly to our second section, neurodegeneration. So why is the brain so uniquely dependent on this? I mean, all cells do it, but why does the brain seem to crumble the fastest when autophagy stops working?
It really comes down to the fundamental nature of neurons. Most of the cells in your body, your skin cells, the lining of your gut, they turn over constantly. They're always dividing. If a skin cell gets full of trash, it can do one or two things. It can divide, which dilutes the trash between two new daughter cells.
Spreading the problem around.
Spreading the problem around, yeah. Or it can just die and be replaced by a new one. No big deal.
But neurons don't divide.
Right. Neurons are postmitotic. For the most part, the neurons you have now are the same ones you had as a child. They have to live for 80, 90, maybe 100 years. They cannot dilute their trash by dividing. They have one option and one option only. They must clean it up. So if the recycling plant fails in a neuron, that neuron turns into a hoarding house, and it's a fatal condition.
And that hoarding is what we see in something like Alzheimer's disease.
That's the idea, but the story is changing in a really dramatic way. And I have to pause here because the source material we looked at regarding Alzheimer's, it highlights a theory that completely flipped my understanding of the disease on its head.
You're talking about the inside-out theory.
Yes. For decades, the popular narrative, the one I always see in the news, the one that drives most drug development, is that Alzheimer's is about gunk building up between your brain cells.
The beta amyloid plaques.
These sticky plaques, right, like sludge clogging up a drain from the outside.
That's the classic explanation. Kill the plaques, cure the disease.
Exactly. But this new research suggests that it isn't the cause at all. It's the result. This theory states that the whole drama starts inside the neuron. The neuron is trying desperately to clean up these precursors to amyloid, specifically a little fragment called APP beta CTF. It's doing its job. It's using autophagy to engulf them, bag them up, and send them to the lysosome for destruction.
To the recycling center we just talked about.
Right, but the recycling center fails. This particular type of trash is too sticky, it's too tough to break down. The lysosome gets stuffed full of this undigested junk till it literally cannot function anymore. It loses its acidity, it swells up, it's completely backed up.
It's like a stomachache that never, ever ends.
It's fatal indigestion for the cell. These failed lysosomes, they're called autophagic vacuoles, just pile up, packing the cell body tighter and tighter. And researchers have a name for this visual pattern. They call it panthos.
Anthos.
It stands for poisonous anthos, which is Greek for flower, because under a microscope, these dying neurons look like beautiful but deadly flowers.
Poisonous flowers. That's a haunting image.
It really is. You see this central core just packed with these failed, swollen trash bags. Eventually, the pressure inside the neuron is just too much. The cell membrane bursts.
And the cell dies.
The cell explodes.
Yeah.
And what is left behind when it dies, that big, sticky lump of undigested trash that was inside it, that is the plaque.
Wait a minute. So you're saying the plaque isn't the weapon attacking the brain from the outside. The plaque is the tombstone of a neuron that has already died.
That is the aha moment. The plaque marks the spot where a neuron died because its internal recycling plant failed. The disease starts inside. With a failure of autophagy, the plaque is the wreckage.
That completely changes how I picture Alzheimer's. It's not just gunk accumulating. It's a fundamental machinery failure.
It is. And we see a similar mechanical failure in Parkinson's disease, but with a much more specific focus. Parkinson's is largely understood now as a failure of mitophagy.
Mitophagy. So that's eating mitochondria, right? The power plants of the cell.
Exactly. And mitochondria are, well, they're volatile. They handle high-energy electrons. They're constantly producing energy, and they wear out. A damaged mitochondrion is incredibly dangerous. It starts leaking toxic free radicals, these reactive oxygen species that damage DNA. So the cell has a very specific protocol to get rid of them.
How does it know which ones are broken and which ones are still good?
It's a really clever flag system. When a mitochondria gets damaged, it loses its electrical charge across its membrane potential. And when that happens, a protein called plen-NK1 starts to accumulate on its outer surface. It can't be cleared away. So it's like putting a bright red flag on a broken machine that says defective, out of service.
And who sees that flag?
An enzyme called parkin. Parkin is the quality control inspector. It sees the PNK1 flags, it flies over, and it coats the entire mitochondrion in these little tags called ubiquitin. It's essentially tagging it for destruction. Then the big machinery, the autophagosome, comes and eats the whole thing.
And in Parkinson's, this handoff from PNK1 to Parkin breaks down.
It breaks down. Many cases of familial or inherited Parkinson's are caused by mutations in the genes for PNK1 or Parkin. The flag never goes up, or the tagger never shows up to the job. So you end up with neurons, especially the dopamine-producing ones in the substantia nigra, that are full of these rotting, leaking power plants.
And the source material mentioned a gender disparity here as well, with another gene, which I found really interesting.
Yes, with a gene called LRRK2, pronounced LRRK2. Mutations in LRRK2 can also jam this whole process. And what's fascinating is that men carry these mutations more often, but the women who do have the mutations suffer a much faster progression of the disease and worse symptoms.
Do we have any idea why?
The leading hypothesis is that it has something to do with how estrogen interacts with lysosomal function and mitochondrial health, but it's an active area of research. It just highlights that autophagy isn't one size fits all. It varies by sex, by genetics, by cell type.
So in the brain, it seems very clear that autophagy is the hero.
Right.
We want more of it. If we can boost it, we might be able to save these neurons. But then we get to cancer, and suddenly the whole narrative gets, well, it gets really complicated.
This is the double-edged sword concept. In the context of cancer, autophagy is a shapeshifter. Is it a hero or is it a villain? And the answer is, it depends entirely on the timeline.
Okay, let's break that timeline down. So phase one, before you have cancer in a healthy person.
In a healthy cell, autophagy is a tumor suppressor.
Mm.
It is unambiguously the good guy. It's constantly cleaning up damaged DNA. It's eating those broken mitochondria we just talked about that produce mutation-causing free radicals.
Yeah.
It's maintaining genomic stability. It prevents the cell from ever turning malignant in the first place.
And we have direct proof of this. It's not just a theory.
We have very strong proof. The gene Beclin-1, which we mentioned is crucial for starting the whole process, is found to be missing or mutated in 40% to 75% of human breast, ovarian, and prostate cancers. When you lose the ability to recycle, you are far more likely to get cancer.
Okay, so we definitely want autophagy running to prevent cancer from ever starting. But what happens once a tumor is already there? Phase two.
Once a tumor is established, autophagy switches sides. It becomes a tumor promoter.
But why? Why would it help the enemy? That seems so counterintuitive.
Think about the life of a tumor cell. It's growing uncontrollably. It's chaotic. It very quickly creates a hostile environment for itself. It runs out of oxygen, which is called hypoxia, and it runs out of nutrients because it has outgrown its own blood supply.
A normal cell will just die in those conditions.
A normal cell would absolutely die. But the cancer cell is smarter. It ramps up autophagy. It goes into survival mode.
It starts eating itself to survive the starvation it created.
Precisely. It recycles its own parts to get the energy it needs to keep dividing. It uses autophagy to become dormant and resilient against these harsh conditions. And this is the real kicker. It uses autophagy to survive chemotherapy.
Of course, because chemo is a massive, massive stressor on a cell.
It's designed to be. Chemo damages the tumor's DNA, its organelles. The tumor says, ouch, huge damage. That's a stress signal. Turn on the recycling plant, fix the broken parts, and keep going. This is a major, major cause of therapeutic resistance in cancer treatment.
So in that case, if you have an active tumor, you might actually want to stop autophagy to make the chemo more effective.
Precisely. And that's exactly what researchers are trying to do in clinical trials. They're using a drug called hydroxychloroquine, or HCQ.
Wait, isn't that the old malaria drug? And the drug that was in the news a lot a few years ago for other reasons.
That is the one. Politics aside, biologically, it works by accumulating inside the lysosome and raising the pH. It makes the acid less acidic. So the autophagosome, the trash bag, arrives and tries to fuse, but the incinerator won't turn on. The trash just piles up and up and up until the cancer cell chokes on its own waste and dies.
And is it working in cancer patients?
The results are promising, but still mixed. In melanoma, one trial showed an 85% response rate when HCQ was combined with other drugs, which is huge. It's also showing potential in very tough cancers like pancreatic cancer. But the problem is that HCQ is a relatively weak inhibitor. We need better, more specific drugs to really jam the gears of the tumor without hurting the healthy cells in the rest of the body.
It's such a delicate balance. You want autophagy to prevent the cancer, but you want to stop autophagy to kill the cancer once it's there.
Which is why the blanket statement, more is better, is never ever the right answer in biology. It is all about context.
Speaking of context, let's pivot to the stuff our listeners can actually control. The lifestyle interventions. Everyone wants to know, what can I do right now to keep my cellular gears clean? And the source is pretty clear on what the gold standard is.
The gold standard for longevity across almost every animal model we have ever studied is caloric restriction, or CR.
And we're talking about a significant cut here, aren't we? This isn't just skipping dessert.
No, this is a 10% to 40% reduction in total calorie intake, sustained over a lifetime, but without causing malnutrition. You still get all your vitamins and minerals in mice, flies, worms. This robustly extends life. And the mechanism is largely understood to be through the induction of autophagy. You create a chronic, mild energy deficit, which keeps AMPK active, keeps mTOR low, and the repair systems stay humming along.
And I think I speak for myself and probably many listeners here, cutting my calories by 40% for the rest of my life sounds absolutely miserable. I like food way too much.
It is remarkably difficult for humans to maintain long-term, which is precisely why intermittent fasting, or IF, has become so incredibly popular. It tries to hack the system. Instead of chronic, day-in, day-out starvation, you cycle it.
You get the benefits without the constant misery.
That's the goal.
So how long do you actually have to fast to turn this on? Because I see people doing 12-hour fasts, 16 hours, 24 hours, even longer. Is there a magic number?
Well, it's not a digital on-off switch. It's more like a dimmer dial that you turn up.
Yeah.
But the science suggests that for most people, at around 12 to 24 hours of water-only fasting, you finally deplete your stored glycogen, that's your sugar stores, in your liver, and that is the wall.
The glycogen wall.
Once you smash through that wall, your insulin levels plummet, another hormone called IGF-1 drops, and the cell finally says, okay, no more food is coming in, time to switch to our internal reserves. That is the point when the autophagy flux really starts to ramp up significantly.
But there's a warning here in the notes, too. Prolonged starvation isn't a free lunch, so to speak.
No, absolutely not. If you push it too far, we're talking multiple days and days without food, you risk excessive depletion. You can start breaking down essential tissue like heart muscle. And in older adults who may already have compromised lysosomes to begin with, too much stress can cause the system to collapse rather than clean up.
So balance. Again, it always comes back to balance. What about exercise? Can I just run instead of fasting?
To a degree, yes. High-intensity interval training, HIIT, and resistance training are both potent autophagy inducers.
How does that work? Is it the same mechanism?
It's the same fundamental energy crisis, just localized. When you sprint or lift a heavy weight, you rapidly burn through the ATP in your muscles. That immediate spike in AMP activates AMPK right away. It also causes a spike in calcium levels inside the cell, which is another powerful trigger for autophagy.
So exercise basically mimics the starvation signal, but it's happening locally in the muscle that's working.
Exactly. It forces the muscle to clean up its proteome, to recycle damaged proteins, which is what maintains muscle quality over time. This is a huge reason why exercise is so crucial for preventing sarcopenia, or age-related muscle loss. It keeps the quality control systems running at full tilt.
Okay, so eat less, fast occasionally, lift heavy things, that's the hard way. But what if we could just take a pill? This is the whole frontier of mimetics drugs that trick the body into thinking it's fasting. And there is one absolute superstar in this field.
Rapamycin.
This drug has such a fascinating backstory. It was found in the soil of Easter Island, Rapa Nui, correct?
Yes, in a soil sample taken from under one of the giant moai statues back in the 1960s. It was originally discovered as an antifungal agent, but in the longevity world, it is the undisputed heavyweight champion.
Why? What does it do that's so special?
It is the single most effective compound for lifespan extension we have ever found in animal models, period. It works by doing exactly what its name suggests. It is the mechanistic target of rapamycin. It inhibits mTOR. Remember, mTOR is the growth, don't recycle, signal. Rapamycin binds to it and shuts it up.
So the cell thinks, oh, we must be fasting, and it turns on autophagy even if you just ate a cheeseburger.
Precisely. It unblocks the pathway. But until very recently, almost all of our data was on mice. That's why the Pearl trial has such a landmark study.
Let's dive into Pearl. This was a human trial, right?
Yes, it stands for Participatory Evaluation of Aging with Rapamycin for Longevity. They took 114 healthy adults aged 50 to 85, and they gave them a low dose of rapamycin, either 5 milligrams or 10 milligrams, once a week for nearly a year.
Okay, so what did they find? First off, was it safe? Because at high doses for transplant patients, rapamycin is a powerful immune suppressant.
That was the #1 concern. But at this low, pulsed weekly dose, it was found to be very safe. The side effects were basically indistinguishable from the placebo group.
That is a huge hurdle to clear right there. What about the benefits? Did it work?
This is where it gets really interesting. The benefits were strongly sexually dimorphic.
Meaning that men and women responded differently?
Significantly differently. The women in the 10 milligram group saw a statistically significant increase in lean muscle mass. They also reported significantly less pain across their bodies. The men, they really didn't show nearly as strong of a response.
Wow. That is fascinating. Do we have any idea why?
We don't know entirely yet, but it actually mirrors what we see in mice. Female mice often get a bigger longevity boost from rapamycin than male mice do. It likely has to do with baseline mTOR activity and how it interacts with hormones like estrogen and testosterone.
Now, there was also a really scandalous, or at least shocking finding about the drug itself in this trial, something that felt like a huge buyer beware moment for anyone in this space.
Oh, the compounding issue. This got everyone completely off guard.
So for the listeners, rapamycin isn't exactly something you can pick up at CVS easily for anti-aging. It's an off-label use. So many people go to compounding pharmacies.
Right, you go to a specialist pharmacy, they take the raw powder, they mix it, put it in a capsule for you and sell it to you. It's often cheaper and it's custom made.
And the Pearl trial used these compounded capsules for a huge chunk of their participants. But when they checked the blood work, what did they find?
It was almost tragic. The people who were taking the compounded pills, the drug wasn't getting into their blood.
At all.
Barely. The commercial Big Pharma version, which is called Rapamune, has this very specific patented nanocoating technology to help it survive the harsh acid of the stomach. The raw powder and the compounded stuff didn't have that. As a result, it was 3.5 times less bioavailable.
Wait, 3.5 times. So if I'm taking a 5 milligram pill thinking I'm extending my life, I'm actually only absorbing, what, maybe 1.5 milligrams?
Or even less. You're basically taking an expensive placebo.
That is a massive finding, because how many people listening right now are spending hundreds of dollars a month on compounded rapamycin?
Probably a lot of them. And this trial strongly suggests that unless you have that very specific pharmaceutical coating, you might just be making expensive urine. It's a huge quality control red flag for the entire longevity supplement industry.
Wow. Okay, so rapamycin is the king, but you have to get the real stuff. But the source material introduces a partner, a required sidekick, spermidine.
Yes, spermidine is a polyamine. It's found naturally in things like wheat germ, aged cheese, and soy.
And I've seen supplements for it everywhere lately. But the source says it's not just another option on the menu, it's actually required for rapamycin to even work.
This is a relatively recent and breakthrough discovery. We used to think they were just two different roads leading to the same destination. But it turns out that there is a deep synergy. Rapamycin gives the signal, go recycle. But spermidine provides the actual tools to build the recycling machinery.
Okay, how does it do that? What's the mechanism?
It does it through a process with a fantastic name, hypusination.
Hypusination. That is a great Scrabble word.
It is. Spermidine is used to modify a specific translation factor in the cell called EIF5A. And when EI5A is hypusinated, when it's activated by spermidine, it gains the unique ability to translate certain very difficult proteins. And one of the key proteins it translates is TFAB.
TFEB, that's the master planner we talked about earlier, the architect who builds the lysosomes.
Exactly. So here's the connection. Rapamycin unlocks TFAB by inhibiting mTOR. It opens the door for him to go to the nucleus. But spermidine ensures that TFAB actually gets synthesized in the first place so it can walk through that open door and do its job.
So if you take rapamycin but you're deficient in spermidine from your diet or supplements,
You might give the signal to recycle, but you can't build the incinerators to do the job. The blueprints are there, but the construction crew never shows up. It strongly suggests that a stack of these interventions might be necessary to get the full effect.
that connects the dots so perfectly. It's not just about one silver bullet, it's about making sure the whole supply chain is working.
The entire assembly line has to be functional.
Okay, so we've covered the engines, the diseases, the lifestyle, the drugs. But we promised to talk about the dark side. We mentioned cancer, but there is also a risk of just over-recycling. Can you do too much of a good thing?
You absolutely can. And it has a name, autosis.
That sounds terrifying.
It is terrifying on a cellular level. It's a specific form of cell death that's driven by the Na plus K plus ATPase pump, the sodium-potassium pump in the cell membrane. Basically, if you drive autophagy way too hard through extreme starvation or by using very aggressive research peptides, the cell eats its own organelles too fast. The nucleus caves in on itself, the organelles just vanish, and the cell dies.
The factory dismantles itself while the workers are still inside.
That's a vivid but very accurate analogy. This is why we have to be so careful with this idea that more is better, especially in acute, high-stress contexts like ischemia from a stroke or a heart attack. In those moments, the cells can panic, they can overactivate autophagy, and they can kill themselves via autosis. In those specific moments, blocking autophagy can actually save tissue.
Which brings us to what feels like the biggest problem in this entire field. How do I know if I'm in the Goldilocks zone? How do I know if I'm recycling just enough or way too much or not at all?
The measurement problem, yeah. For a long, long time, we were flying completely blind. We had to rely on these static markers, like taking a snapshot of those LC32 levels we mentioned.
But that's tricky, right? Because you said a high level of LC32 could mean a lot of recycling is happening. Or it could mean the incinerator is broken and the trash bags are just piling up.
Exactly. It's the traffic jam analogy. If you look at a highway from a helicopter and you see 1,000 cars, does that mean traffic is moving really, really fast, high flow? Or does it mean there's a roadblock up ahead and no one is moving at all? A total blockage. A single snapshot cannot tell you the difference. You need to measure the flux, the actual speed of the cars.
So how are we solving this? How do we measure flux?
The new frontier is something called whole blood flux assays.
Okay, how does that work?
You take a blood sample from a person, you split it into 2 tubes, one half you leave alone. The other half you treat with a blocker, like chloroquine, that drug that blocks the lysosome and raises the pH.
You create the roadblock.
You intentionally create a traffic jam in the second tube. Then you measure the LC3 markers in both tubes. The difference in the amount of LC3 that builds up between the tube with the block and the tube without tells you how many cars were actually moving down the highway.
Ah, I see. So if the blacked tube has way more cars piled up than the normal tube, it means the normal tube was clearing them effectively. The flow was high.
Exactly. That difference is your autophagic flux. And what researchers are finding is that this flux varies wildly between people. It varies by cell type. It seems monocytes in the blood are the most sensitive, and it varies by your diet, your genetics, your age.
So the future is, I go to my doctor, they draw my blood, they run a flux assay, and they can tell me, hey, your recycling plant is running a little sluggish. You probably need to fast for 16 hours twice a week and maybe consider 5 milligrams of rapamycin.
That is the definition of precision medicine. Instead of generic advice for everyone, we tailor the intervention to your specific measured cellular capacity. We are not there yet for the general public, but that is where the science is headed, and it is headed there fast.
And that leads us to the final big takeaway from all of this, the concept that ties it all together, hormesis.
This is the philosophy that makes sense of it all. We do not want constant chronic autophagy. That leads to muscle loss, sarcopenia, and potentially autosis. We also do not want constant chronic growth from mTOR. That leads to cancer and clogged senescent cells.
We want the rhythm.
We want the pulse. A period of stress followed by a period of recovery.
Yeah.
Fasting followed by feeding. Intense exercise followed by rest and rebuilding. That oscillation, that rhythm is what keeps the entire system robust and resilient.
It's a beautiful thought. You know, we often think of aging as this slow, inevitable decline, like a battery just running out of juice.
But the science really suggests it's more like a mechanical failure that comes from a lack of maintenance. It is a slow, steady clogging of the years.
And if we can master that rhythm, the rhythm of eat versus repair, of mTOR versus AMPK, we might not just live longer. We might be able to prevent the very structural collapses that we call Alzheimer's or cancer. We might keep the factory running smoothly right up until the very end.
And that is a goal worth skipping a meal for now and then.
Absolutely. So for everyone listening, here's your final thought to take with you. Look at your own life. Are you stuck in growth mode? Constant snacking, constant sitting, constantly stimulating mTOR? Or are you giving your cellular factory the downtime it needs to clean the floors?
The power to clean house is literally inside you. You just have to give it the right signal.
Thanks for diving deep with us. We'll catch you on the next one.
Stay curious.